Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today, we're going to talk about bacterial structure and classification. So bacteria are unicellular prokaryotic organisms that are incredibly abundant and diverse. There's an estimated 5 times 10 to the 30 bacteria on Earth. And in the human body, bacterial cells outnumber human cells by about 10 to 1. Bacteria can live independently, but many form relationships with other organisms. The first is mutualistic symbionts. These provide a benefit to the host. An example of this is the bacteria in our gut. These aid digestion and produce vitamins K and biotin, which is B7. They also prevent the outgrowth of harmful bacteria. Babies are actually given a vitamin K injection at birth because they are not yet colonized by bacteria yet and cannot produce clotting factors, and they are at a risk of hemorrhaging. There are also pathogens. These cause disease by invading tissues or producing toxins. And finally, you have parasites. These live off the host without providing a benefit and often harm it. Bacteria are generally classified into 3 main shapes. You have coccus, which are spherical, like Staphylococcus. You have bacillus, which are rod-shaped, like Escherichia coli or E. coli, and you have spirillum. These are spiral-shaped, like Helicobacter pylori or H. pylori. Now, let's talk about bacterial cell structure. All bacteria have a nucleoid region, ribosomes, cytoplasm, and a plasma membrane. So start with the plasma membrane. This is a phospholipid bilayer with embedded proteins, and it controls transport into and out of the cell. The cytoplasm is a gel-like fluid that fills up the cell and contains all the essential components for growth, metabolism, and replication. Since prokaryotes don't form multicellular organisms, each bacteria is responsible for protecting itself from the environment. The cell wall is the outer barrier of the cell. It provides structural support, protection, and is made of peptidoglycan. This is a polymer of sugars and amino acids. It even contains D-alanine, which, as you may recall, is not found in animal cells. It helps maintain shape and controls the movement of solutes in and out of the bacteria, and this allows the cell to maintain concentration gradients. The cell wall and plasma membrane together are known as the envelope. The cell wall is a target of many antibiotics, including penicillin. The enzyme lysozyme, which is found in tears and saliva and is made by lytic viruses, destroys the peptidoglycan in the cell wall, resulting in a protoplast, which is osmotically fragile structure. So prokaryotes concentrate their DNA in a nucleoid region, but they don't have a nuclear envelope. Prokaryotic DNA is carried on a single circular chromosome, and in bacteria, this DNA is not coiled around as histones, unlike archaea and eukarya. Plasmids are small circular DNA, and this DNA is not essential for survival, and this is why it's not considered part of the genome, but it may give an advantage like antibiotic resistance. Plasmids can be transferred between bacteria via horizontal gene transfer. Let's move on to flagella. Bacteria can be monotrichous, which means you have flagella at one end, amphitrichous, which means flagella at both ends, or peritrichous, which means you have multiple flagella. Flagella is a long whip-like appendage, and it's used for motility. It rotates like a propeller and is powered by a proton gradient. This allows bacteria to move toward nutrients or away from toxins. This is called chemotaxis. The flagella is composed of a filament, a basal body, and a hook. The filament is a hollow helical structure composed of flagellin. The basal body anchors the flagella to the cytoplasmic membrane and is the motor of the flagellum. The hook connects the filament to the basal body, so as the basal body rotates, it exerts torque on the filament, which can spin and propel the bacterium forward. So prokaryotes don't have a mitochondria, and the cell membrane is actually used for ATP generation and for the electron transport chain. Prokaryotes also contain ribosomes, but the ribosome is a different size than the eukaryotic ribosome. The eukaryotic ribosome has a 40S and a 60S subunit, and the prokaryotic ribosome has a 30S and 50S subunit. Some bacteria also have the capsule or glycocalyx. This is a sticky layer of polysaccharide goo surrounding the bacterial cell and often surrounding an entire colony of bacteria. It makes bacteria more difficult to be eradicated by the immune system. It allows bacteria to adhere to smooth surfaces like rocks in a stream or the lining of the human respiratory tract. Streptococcus pneumoniae has a capsule which allows it to survive in the human respiratory system, and it in turn causes diseases like pneumonia, sinusitis, meningitis, and otitis media. Pillae are long projections on the bacterial surface and are involved in attaching to different surfaces. The sex pilus is a special pilus attaching F+, which is male, and F-, which is female, bacteria. This facilitates the formation of conjugation bridges, and we'll talk about this in a future episode. Finally, they have fimbriae. These are smaller structures that are not involved in locomotion or conjugation, but they're involved in adhering to surfaces. Now, there are two types of cell wall, gram-positive and gram-negative. The type of cell wall can be determined by gram staining, which uses a crystal violet stain followed by a safranin counterstain. So gram-positive bacteria have a thick peptidoglycan layer, they have an absent outer membrane, they have liptoteichoic acids, but they have a minimal periplasmic space, and the stain color will be purple. Gram-negative bacteria have a thin peptidoglycan layer, the outer membrane is present, they don't have liptoteichoic acids, they have a prominent periplasmic space, and the stain color is pink or red. So gram-positive bacteria, thick peptidoglycan layer, this retains the crystal violet stain, which is purple, and the liptoteichoic acid helps the bacteria evade the immune system and contribute to the development of certain infections, and it also activates the human immune response. Gram-negative bacteria have a thin peptidoglycan layer, it does not retain crystal violet, and instead takes up the safranin, which is pink. The outer membrane contains lipopolysaccharides, which is LPS for short, and these act as endotoxins. Lipopolysaccharides are part of the bacteria that trigger the immune response in humans, and the immune response to lipopolysaccharides is much stronger than the response to liptoteichoic acid. The periplasmic space is the region between the outer membrane and the plasmamembrane, and it contains enzymes for nutrient processing and antibiotic-resistant factors. Overall, bacteria are structurally simple, but they're incredibly diverse in their form, function, and ecological roles. Their cell envelope, plasmids, and appendages like flagella are key to survival, reproduction, and adaptation in nearly every environment on Earth. That's it for today. Next time we'll go over bacterial growth.